Colloquium. It's a great pleasure to have today Silvina Ponce Dawson from CONICET, from UBA, and many other institutions, as you can see. So let me introduce our speaker. So she got both the licenciatura and the PhD degree in University of Buenos Aires, and then she had a postdoc career in US, in University of Maryland for a couple of years, then other 3 years in Los Alamos, and then she came back to Argentina and to Buenos Aires, where she did all the professorship career, and she became full faculty in Buenos Aires in 2017. Among other, let's say, institutions, she's currently the president of the International Union of Pure and Applied Physics, the UPAP, and she will run this union up to 2027. She will be our visitor for the entire 2025 year, so we will have plenty of time to discuss and to enjoy time with her. And she's an expert in statistical physics and applications to biology, and today's talk is about that, and this talk is named Amplitude and Frequency Encoding in Cell Signaling. Silvina, the floor is yours.
Thank you for the invitation. So let me see how this works. No, this is the way, oops, I don't know what that is. How do I go back? Oh, I see. That's it. Okay, so now I've added UNESP to my affiliation, and so I'm thankful to Nathan for the invitation to spend here 10 months, and to FAPESP for the funding to do that. And today I'm going to talk about amplitude and frequency encoding in signaling, which is work we did with Alan Gibré and Alejandro Coleman-Lerner. Alejandro is a biologist. Alan was a student we co-advised his PhD. Anyway, so this is about how cells interact with their environment to generate responses. And usually what happens is some substance changes its concentration. It could be that it was absent and suddenly it appears, or it can be that it changes its concentration in the environment. And so the cells detect those changes by binding those substances, those molecules, to receptors that are on the plasma membrane, and that induces changes in the protein. And so here is the plasma membrane receptor, is this wiggly thing here, which crosses the membrane. And so on one side, it binds the molecule whose concentration changed. And then on the other side, something happens to, usually these are proteins, and to the conformation of the protein, and that induces a sequence of changes in what is called a signaling cascade. And eventually, that signaling cascade can result in changes in gene expression. Basically, this is a scheme for an eukaryotic cell where you have the DNA, most of the DNA in the nucleus, and so something enters in the nucleus and modulates the gene expression. Now, so that's a little bit of what I said. Usually, the modulators of gene expression are called transcription factors that need to bind to certain regions of the DNA to let there is an enzyme, a polymerase, that reads the genetic information and brings together, puts together the amino acids that are going to make the messenger RNA that is then going to be translated into the proteins that are in that cell. But for the polymerase to know where to start reading, so which genes should be read, there is something that has to be bound to a specific sector, so that it's like a signal that tells the polymerase you should start here to read. And so this is a very, very simple scheme. Then I'm going to show a little bit more realistic one from a book by Bill Bialek, where the transcription factors bound and like helping the polymerase start the readout of the genetic information. Anyway, so the gene expression you can change by changing the transcription factors, which are very specific, some of them inside the nucleus in the case of these type of cells. Okay, so one cell type in which many of these signaling processes have been studied is the yeast cells, basically because they are cheap and they are easy to manipulate. And my colleague Alejandro, with whom we worked, he works on yeast. And so what they do is they use different techniques to change the proteins to add them like a fluorescent tag somehow. And so then you can use microscopy techniques to observe changes in the presence of the proteins. And sometimes when you subject those cells to a change in the environment, which I told you could be the change of the concentration of some substance, maybe you produce a stepwise change in that concentration, and you see that the signaling cascade that follows is characterized by the increase in the concentration of other substances, and that increment in the concentration follows more or less the same dynamics as that of the external input, which is that concentration that changed stepwisely. But in other cases, the intermediaries show a non-trivial dynamics, a dynamics that does not mimic at all that of the external effector. And so many times what you observe is pulsatile behavior. That's the frequency of the title. And so in this paper from 2014, these people looked at the transcription factors in the nucleus of yeast cells, many of them, and of all those that they followed, what they did was subject the cells to some sort of stimulus that would induce the entrance of the transcription factor that's specific to the response related to that stimulus, and they monitored how was the entry of the transcription factor into the nucleus, and they observed in 8% of the transcription factors that they looked at, they observed this pulsatile behavior. And now why do cells use this strategy? And when is it most appropriate? Because as I told you, it's not always the case that there is this non-trivial dynamics. And does this improve the cell signaling capabilities? We might think that you're going digital, no? You have pulses and maybe is more resilient to noise. Anyway, so that was a question that motivated our work. And let me show you one example in yeast again. This is another group. It's a group led by Erin O'Shea. And what they did was they subject the cells to different types of stresses. So 3 types. One was glucose limitation, and another was osmotic stress. The other one was oxidative stress. And what they observed was that the response that the cells generate to each of these stresses are mediated by the entry into the nucleus of the same transcription factor, but with a different dynamics. And so in the case of the glucose limitation is 1 pulse of nuclear factor in the nucleus followed by many pulses. In the case of osmotic stress, is an initial pulse and no more pulses after that. And in the case of oxidative stress, is 1 prolonged elevation of the transcription factor, which is called MSN2, by the way, in the nucleus. So these are the plots they produced. And let me show you some things that are interesting. For example, here in the case of osmotic stress, what they saw was that that the duration of that pulse as a function of the level of the stress was an increasing function. So there you talk about you're encoding the level of stress into the duration of the increment of the transcription factor in the nucleus. In the case of oxidative stress, it's the what's the amplitude, what's the concentration of the transcription factor in the nucleus. That's the increasing function of the level of stress. And in the other case, which is glucose deprivation, then it's the frequency of those bursts of the localization of the transcription factor in the nucleus that increases with the level of glucose deprivation. Oops, here. So we have 3 types of encoding with the same transcription factor. So in this case, the dynamics is used to use the same transcription factor to detect different types of stresses and, of course, generate different types of responses. So now this is the example that I'm going to look in more detail, which is, yes.
There's so few points and so huge error bar.
In the experiments, it's not so easy to do the experiments. Oops, I don't go, oh, no. Here. This is the number of experiments the number of situations that they analyze. Those are 4 situations that they analyze, the 4 levels of stress that they analyze. What I don't remember is how many cells they looked at. So the error bars are related to how many observations they got for that particular condition, but they looked only at 4 different conditions. That's, I guess, because of problems with it's not so easy to do the experiments.
Just out of curiosity.
Yes.
Are there other groups that try to reproduce this, or is it an
This
important result?
Oceas, this thing about the, in this part, well, they did lots of work. I don't collaborate with them. It's a motivation for the study. First, they observed something that I think is very interesting, which is that you can use the same transcription factor to generate different types of responses to the same. And then what they did was other experiments in which they manipulated themselves the transcription factor. Because
I'm asking a different question. So if a physicist finds some surprising result, usually other physicists try to reproduce it. Are there any other groups that tried to reproduce what they found?
I don't remember if exactly this type of experiments. I don't remember,
okay,
really.
Thanks.
I really don't remember. Anyway. But what they did was then, before they observed, it would be more or less physiological conditions in the sense that you produce the stress and see the response. But then they did experiments in which they manipulate the transcription factor themselves so that they can manipulate the dynamics of how it enters into the nucleus, and they reproduce the, they were all consistent with their own.
So to understand, I'm here.
Yeah.
At the very end, the cell is producing exactly the same protein because the transcription factor is the same, right? But at different rates depending on how...
Here there are 2 types of proteins, let's say. One is the transcription factor that here, the role that it plays is to modulate gene expression. So
exactly.
then you are going to produce some other protein,
exactly,
which is related to the response.
Right, but in the 3 cases, you produce exactly the same end result, but with different rates.
No, the end result is not. What it is that you have the stimulus and and you're going to change the production of some proteins, and who is going to tell the cell that they have to change the production of those proteins is the same transcription factor. What it changes is the dynamics with which it enters into the nucleus. Okay, anyway, so this is the case that I'm going to look at in a little bit more detail. This is these yeast cells, usually they just divide. But in some occasions, particularly when they are deprived of nutrients, they become in 2 types, with 2 different types. And each subtype of these yeast cells secrete a pheromone that attracts the cells of the other type. And so when that happens, and you are, for example, you are some type which secretes a particular pheromone that attracts the other. What induces is a polarized growth. So this is shown here in this scheme that I took from that paper. There is the two types of cells. They secrete the pheromones. This polarized growth starts, and then they eventually fuse. And they mixed up their genetic information. Then they sporulate, and they propagate the species. So it's a way to survive in not very nice conditions. It's common of these type of organisms. And it's used to understand the transition to sexual reproduction. And anyway, so there have been many works trying to dissect the whole sequence of reactions in the signaling cascade that goes from changing the pheromone to the growth of this protrusion that is called shmu. And of course, you use fluorescent things. And in the experiments, you cheat the cells. Instead of putting in a potential partner, what you do is you put the pheromone and expect that the cell will respond as if there was a partner there. Anyway. So I'm not going to look at all this detail of the pathway. That I don't know all the details very well either. And so you have the pheromone attached to the receptor, and then a sequence of reactions and then changes in gene expression. And that leads to this polarized growth, and which is towards the region where the pheromone is larger. Basically, if you put a gradient of pheromone, the growth is going to more or less go to the region of larger pheromone. It also depends on the initial state of the cell. Anyway, so in this case, you need to sense where the pheromone is larger. So you need to be able to distinguish different levels, different concentrations of pheromone. And one of the questions that we ask is how well they can detect these changes in pheromone concentration. What are the smallest values that can be detected? And so you can subject the cells to different concentrations of pheromone and see what happens. Something that happened was that in the presence of pheromone, you have what we call the amplitude encoding response type, which is that the intermediaries of the cascade, they show a dynamics that is similar to that of the change you introduced in the pheromone, which is not pulsatile. And in other cases, you can have also pulsatile behavior. And so before, we saw that the pulsatile behavior was used to multiplex, so to use the same signaling molecule, the same transcription factor, with different dynamics to signal different stimuli, to identify different stimuli. In this case, it's one type of stimulus, and then for the response that you are going to generate, you have some part of the pathway that is pulsatile and some other part that, because the whole response is complicated, you have to grow, and then you have to break these cells have a wall, you have to break the wall to be able to fuse and stuff like that. But some parts of the whole response are mediated by a pulsatile intermediary. So it's a different thing, and we ask why that happens. And so let me go a little bit more in details of this example. So these are some experiments in which they observe some participants in this signaling cascade, and one of them, as a function of the concentration of the pheromone, which is called alpha factor, this is the response level, and one of them is measuring the transcription and induction. And you observe this type of behavior, which is increasing after you cross some sort of a value, and then it saturates the level of response. And And so what you say here is that the level of response is higher, the higher is the level of pheromone. So you have amplitude encoding there. But then increasing pheromone concentrations, they also induce a behavior which is pulsatile in the intracellular calcium concentration. This is an example of the pulses that you observe. And these are the cumulative distribution functions of the number of pulses that you observe in each cell for increasing levels of pheromone concentration, so that you see that you move to the right, which means that you have more bursts elicited when the pheromone concentration is larger. So basically, what we say is that here you have amplitude and frequency encoding. So that's another motivating case for the type of work that we do. And so one way in which you can approach this question, which was what's the difference between encoding in amplitude and in frequency? What are the advantages when one of them could be better suited than the other? We used information theory, and basically we drew inspiration from Bill Bialik's work. And so the idea is to assume that the environment can be in a sequence of states that we call W, and with a certain probability, which is here. And then the cells somehow obtain information on what state the environment is in, or measure or something that would be, for example, the changes in those internal, in the intermediaries of the signaling. In particular, what we look at is at the level of proteins that are created as a response to the external stimulus, to the input. So that measurement or response we call D, and so the mutual information measures how much information, how much you reduce the level of uncertainty with respect to what you didn't know at the beginning, in which state among a possible set of states the environment was, how much it's reduced by measuring, which in this case is by producing a certain protein that you are going to use to generate your response. And so that's what this is expressed here. Here, this is the entropy of the environment. And this is the conditional entropy when you measure some D. But then you average over all possible values of D with their own probability of occurrence. That's basically the mutual information. And, well, and you can put it in terms of the joint probabilities and the marginal probabilities of the things. Anyway, so we use this, and to study the multiplexing, we studied the mutual information between the transcription factor and the messenger RNA produced, integrated over time as an indicator of how much protein you are producing. And in this second work, that is the one that I'm going to talk, is we looked at the whole information transmission from the external stimulus to the production of the messenger RNA. Okay, so we computed this mutual information, and we needed to, this is something that we do numerically, and then we have some analytic results too. But then we need models in between. And so what we used was a different mapping from the external stimulus, which we call IX. That would be the level of pheromone, for example, the concentration of pheromone. It could be something else. And then we had a different type of dynamics of the transcription factor inside the nucleus that we call TF, that transcription factor in the nucleus, different depending on whether it's amplitude or frequency encoding. and then a very simple transcription model from the transcription factor to the production of messenger RNA. And so this is a scheme of the first part, basically. So we have an external stimulus, which is like a stepwise, but we are looking at the time during which the external stimulus is constant. So we assume it changed at some point and then it stayed. And then in the case of amplitude encoding, we assume that there is one pulse of nuclear factor inside the nucleus that lasts 10 minutes, and that there is some mean amplitude of that pulse, which is a function of the external stimulus, very much in the same type as the plot that I showed you of the response as a function of the pheromone concentration, which is a Hill function, something that has a saturating value. It has a value at which it starts to, or value that shows when it reaches kind of mean level between 0 and the saturating value, which is called EC50, and then a Hill coefficient, which is called H, which regulates how sharp that plot is. If it's H larger, is sharper. And plus some noise, which is Gaussian noise over here. And in the case of frequency encoding, then I'm going to explain why we choose that. We choose a sequence of a random sequence of pulses, all of the same mean amplitude plus noise. And the separation, the time separation between successive pulses is a random variable that is exponentially distributed. And the mean time between successive pulses is also an exponential function, well, but I don't know what happens there, of the external stimulus. And I'm going to explain why it's an exponential. Yep. Pardon me? You're trying to model what's going on in the cell? Yes. What we are trying to pick is some judicious, simple model of what happens in the cells, not just in the cell, in the nucleus, as a result of the external stimulus. Yeah, we are going to use the model to compute MI. Then we have now, with a student, we are going to compute from experiments MI. And also the O'Shea, they also computed MI from experiments. In that case, they looked at the protein produced, observing them. They introduced some external stress, and then they looked at the protein produced and computed the mutual information. So you can do it with experiments. We are going to do it with a model.
It's not clear to me why that number is something that's relevant to compare the model with the experiment.
The number itself, I don't think it's that relevant because that is a, there are lots of simplifications. But what it does make sense is to compare, within the same model, the behavior for frequency and amplitude encoding. That's the absolute value that we get, it's similar to those obtained for the experiment. So that was reassuring, but I wouldn't pay that much attention to that, rather to the comparison.
I understand the behavior.
Silvina, sorry, another simple question.
Yes.
I'm here.
Oh, okay. I thought it was somebody, because I was seeing someone there.
If I understand, let's see. The idea is that then the mutual information will play a sort of, let's say, cost function role. So you want to optimize the amount of information that you get in the two cases.
We don't want to see, we are not looking here to optimize anything. We are just seeing how it be. Well, let me tell you what we were after with this. Advance a little bit what I was going to say.
No, okay, let's wait.
But I wanted to see if because in our previous work, we didn't find much difference between frequency and amplitude encoding.
Okay.
And we were looking from the transcription factor to the production of mRNA. And so then I said, well, let's look at the from the stimulus, and maybe frequency acts better for certain ranges of stimuli, and amplitude is
But
better.
better in what? In producing information?
Yes, in producing.
Okay, that
in
was
being
the question.
being able to distinguish
Okay,
levels.
yeah.
Which
That
would
was the
be
question.
comparing the mutual
Okay.
information.
Okay, thanks.
See, yeah, and that's the okay. Oh yes.
So just to make clear, is this through this external stimulus that the cell gains information about the environment?
Yeah, well, that's the thing. What we want to know is the mutual information between that. The external stimulus would be, for example, the concentration of pheromone in the example that I looked, and the protein produced. So whether the protein produced distinguish, so that the values of the protein produced within all the noise that is in between, so that usually it's not that you always produce the same amount in front of the same stimulus, you have a distribution, whether that distribution of proteins that you produce allows you to distinguish different values of the input, that you don't have something that is a mess that, I don't know, that it's like from knowing how much protein you produce, how well can you tell what was the input? That would be a little bit the motivation for this.
Okay, but does the cell gain that information that you said before? Does the cell gain the information about the environment before interacting with it?
Before?
Interacting with it.
Well, no. No, it has to, produce the whole signaling cascade.
Okay.
So you need to bind the stuff.
Okay.
You can also think of other types of stimuli in which you can compute something similar. Here we really are motivated in things that bind and then create this signaling cascade. And this very simple model is like like aggregating many steps of the signaling pathway in a judicious type of dependence between the external stimulus and what we are looking at.
Okay, thank you.
Okay, and this is what I said about the about frequency encoding. And well, maybe we should have included some transient dynamics of the transcription factor getting in the nucleus. But since we are our output is going to be a time integral of the messenger RNA produced, then we think that transient would not affect very significantly that integral, which is our output. Anyway, so then once we have this very rude model of the nuclear transcription factor, then we put this into a transcription model. And the transcription model that we used, we also took from series of models that were analyzed by the group of O'Shea, trying to see which ones reproduced better their results. And this is the simplest among those, which is a model in which the transcription factor can be in two states, P0 and P1. And the transition between the two states depends on the transcription factor in a way which is also hill-like. And then the production of mRNA occurs only when the promoter, this is the promoter, which is the sequence along the DNA where the transcription factor has to bind or has to be in a particular state for the polymerase to know that it has to read the genetic information. And so when the promoter is in that particular state that is favored, the larger the amount of transcription factor, basically, then a mRNA is produced at a rate that also depends on the transcription factor. So this is that description. And we use we abuse the notation in the sense that we call P0 and P1 or P0 and P1 the states and the probabilities that the promoter is in each of the states. Anyway. So, and for the modeling, the numerical part, this transition from the two states of the promoter, we model deterministically. So we have equations for the probabilities, basically. And then the production of mRNA we do with a stochastic equation. So we produce one by one and also degrade, remove the molecules at a constant rate, D2. So one part is fully stochastic, the other is deterministic. And, well, that's the input, the external stimulus. And the output, as I told you, is the integral of the messenger RNA that is produced. And so let me give you an idea of what this very simple model, how you could eventually derive it from a more mechanistic one. And so the next slide is not for you to understand everything. It's just to tell you that the transcription is very complicated. It's not just, it's particularly in eukaryotic cells in which the DNA is all wrapped up and made, I don't know, in a very complicated structure, and then inside chromosomes that need to sort of unbind. And so you need many participants of the cell to be able to, well, unwind the DNA, to open the DNA strands, to indicate to the polymerase. So it's a very messy thing. And let me show you, this is a picture where distant regions of the DNA, distant along the fiber, the DNA molecule, you see, but close from the point of view of space, because everything is all wrapped up, they participate. So it's a very messy situation. And the way in which you can understand that model that was actually obtained from fitting originally, there is a simple way. So this was the model. And what you can think of is that you have like two types of transitions, basically, in this model. One type of transition that depends on the transcription factor that you are interested in. So this is like you bind N molecules of your transcription factor from this state of the promoter to this state, and then you unbind with this rate KB star. But the promoter can be in two types of states before binding or unbinding the transcription factor, which are the ones that don't have the asterisk here. And so you have the same type of transitions here with TF, the transcription factor binding here and unbinding N molecules. And so this would be, these transitions here from the non-asterisk to the asterisk would be all the other mediators of that very messy picture that participate in the regulation of transcription. And this over here would be the binding of the specific transcription factor that we are interested in. And then we assume that transcription proceeds only when the system is in this state. So the promoter is in 4 states, and transcription proceeds when it's only in 1. And if you think that there is and there you produce the messenger RNA with this rate. And so then, this is what I've already said. And assuming that there is a time scale separation, so that the arrows that go horizontal, they occur much faster than those that go vertical. It's hard to keep this in anyway. You have that time scale separation, then it's like you collapse the 2 branches of that plot, assuming that the left and the right branch are in equilibrium with one another. It means that the fraction that you have of the system in one branch is something related to what you have in the other branch. And that's how you come up with this type of dependencies. Anyway, so it's a time scale separation. It's 2 types of transitions 4 states and a time-scale separation, and you come up with this very simple model. Anyway, so we have the how we mapped from a, so let me go a little bit to the difference between amplitude and frequency encoding. For amplitude encoding, we assumed it was a mean that was a Hill function, I told you, and plus noise. And in frequency encoding, it was a weirder dependence with pulses that are stochastic, and the mean, they had a minimum time that has to elapse between 2 subsequent pulses, which is T-mean, plus a stochastic component whose mean is an exponential of the input, the stimulus strength. And that comes, so this is the mean interpulse time. And that comes from, well, these are the rationales. This is what happens when you have reactions, like with enzymes, which is that type of equilibrium relation that I told you about. I don't have time to go through the whole calculation. But basically, if you have this type of binding, oops, here, H molecules, let's say of IX plus the enzyme goes into a complex, but back and forth, then in equilibrium you have this type of dependence of C as a function of the IX, basically. And C here is the amplitude of the transcription factor. Anyway, so let me explain a little bit better the interpulse time, which is the one that is weirder, I would say. It came from observations of intracellular calcium, and which they found that dependence. I'm going to show you some figures. But we also went to a plot of the burst frequency of a nuclear localization of a transcription factor as a function of some external stimulus, which in this case was calcium, external calcium. And we plotted in a log plot. And so we found that there was this dependence as well. So that this is well approximated by an exponential. And so here is the data from the pulses of intracellular calcium in many different cell types that you have that dependence. Here you are plotting not the frequency, but the mean time between subsequent pulses. That's why it's decreasing. Exponential, in the other case, is increasing for the frequency. And, and the way in which you can explain this is that the system has to cross a barrier, like when you have a Kramer's law for reactions, that the time you have to wait for the system to go from one minimum to another minimum goes exponentially with the height of the barrier. And the same thing happens not only when you have a bistable system like here, that you have 2 minima, and the system can go from one minima to another, but something happened with this. But it also happens in excitable systems. And excitable systems is systems in which you have a stable fixed point, you produce a perturbation. If the perturbation exceeds a threshold, then you have a pulse. So if you have an excitable system with noise, every now and then you have a pulse because noise makes the system cross the threshold. And in these intracellular calcium signals, with a very simple model, we could reproduce the exponential dependence. The noise is because the signals are produced by individual calcium entries from individual channels. And that's the noise, and the level of the threshold, how close you are to the threshold, is regulated by this external stimulus. Anyway, so we have that. And then, well, and this is something that is like I would say part of the trickiest part of this whole thing, which is when you compute this mutual information, you need a distribution probability for the stimulus. And if you do experiments, then you control that. if you're thinking of something, I don't know, general, I really don't know how you choose. So since we were after trying to understand whether the response was different depending on the level, the strength of the stimulus, we decided to use a very simple distribution, which is the beta distribution, which is a distribution of compact support. So it's a distribution that is defined between 0 and 1. And it's a, let me look at only this so that I don't make much, because I don't have that much time. So these are the cumulative distribution functions of the beta distribution. It has two parameters, alpha and beta. And so if alpha plus beta is fixed and I change alpha, what I do is change the median, so it moves to the right. So it means that for small alpha, I have medians that are closer to 0. For larger values of alpha, I have medians that are closer to 1. So by changing that, I am concentrating in a distribution of stimuli that is more concentrated around small values or around larger values. That's the aim of this study. And that was what we were after. And so these are the distributions of the inputs. Actually, I'm going to show results with this one here. So it's 10 distributions. And so we have 10 computations of the mutual information, one for each of these distributions. And we are going to plot them as a function. Whoops, this is the thing. Well, this is an interpretation which I'm going to skip here. And so these are the results that we obtained. This is for amplitude encoding. And so these are simulations that we did with that Hill function that we used in amplitude encoding, all of them with the same Hill coefficient by different EC50s. And the different symbols correspond, so each of the curves corresponds to one value of the EC50, and each of the symbols correspond to a different distribution of the input. And so this one over here is what we see is, and we plot them as a function of the median. So this would be for the distribution that is more concentrated into small values of the stimulus, you don't get much information. And the mutual information increases as you increase the level of the stimulus strength for this curve, which has EC50 close to 1. While for this one that has an EC50 much smaller, then you see that this is decreasing with increasing the stimulus strength. So it means distributions that are more concentrated in smaller stimulus strengths have larger mutual information. And this is the same, but keeping the EC50 fixed at 0.5 and changing H. And this is for frequency encoding and different values. And before talking about that, let me wrap up a little bit what we observed for amplitude observation. What we observe is the maximum information, and this is very noticeable when H is large enough, like in this case, the maximum correlates very well occurs when the median of the distribution of stimulus strength is close to the EC50. So it's like amplitude encoding is good at transmitting information over a certain range of stimulus strengths that is related to the rates with which it's going to decode that, which is the EC50 of this transformation. In the case of frequency encoding, there are cases in which the information is relatively flat. It can also change with the medium. but it can also be relatively flat, which is the main message I want to convey. And this comes from the different dependence of how the transcription factor maps the external word. So one thing that we asked was, this is an analytic calculation that is not very complicated, but that shows the qualitative differences between the two types of encoding. And so we asked, what happens if we take two stimuli that are very close together, differ by delta I? How well does the first step in the encoding, the first step that goes from the external stimulus to the transcription factor, how well does it distinguish those two stimuli strengths? And so in the case of amplitude encoding, we have a mean plus Gaussian noise. So what we did was, like when you do a diffraction, grid, basically, which is how well it can discriminate two inputs. It's the ratio between the distance between the two means divided by the width of the distribution, which is twice the standard deviation of the normally distributed random variable there. And then, well, we put the dependence that we have for the amplitude, the mean amplitude as a function of I, and we get this expression. And in the case, and so if this ratio is larger than 1, it means you have the, so you have this thing. This is the idea. So you have, this is the ATF of IX, and this is the ATF of IX plus delta I, and this is sigma, and this is sigma. And so if this difference is larger than 2 sigma, then they are it's easier to discriminate. In the case of frequency encoding, because the mean interpulse time is an exponential, we don't have that nice thing. So basically what we did was you have two exponentials, so what you want is that they are like whatever you have here, that this, most of the, I don't know, anyway, so that when you already go down here you want to be starting to be up, something that they are separated enough. And that you can define in terms of quantiles. And anyway, it doesn't matter how we derive these expressions. You can also obtain through a calculation that is called Kullback-Leibler divergence. But the main point here is that this condition depends on the stimulus strength, and this condition does not. So what it means is that the ability to discriminate two nearby inputs of frequency encoding does not depend on the strength of the stimulus. So basically, it's scale invariant. So this exponential dependence gives you a scale invariance of the discrimination, which in the case of amplitude encoding, that does not exist, because what you do is basically your amplitude, your mean amplitude as a function of the external stimulus is sensitive in this region. And this region is the EC50. And that's why you need to match that to be able to discriminate. In the case of the frequency encoding, you don't need that matching. Of course, then, whole processing, so this is the main message of the talk. And so now this is, you can observe in this thing that I just drew here, but I'm going to skip that. But the main message here is that they are qualitatively different. Of course, then the frequency encoding, those pulses have to be, I don't know, counted or something. Usually what you do is you add the map. And so it depends on the time scales of that subsequent processing, because if your frequency is too slow, you don't have pulses, you cannot count anything. And if your frequency is too large and your processing is too slow, you will also lose information. But if you could just have forever for counting and be super fast, you could discriminate across all stimuli strengths. So what could this be used in the case of the yeast cells and the pheromone response pathway? Maybe it can enlarge the range of, in the case of the pheromone response pathway, of pheromone concentrations over which the cell can detect differences in concentration and make its SHMU grow into the regions of larger concentration. So these are the experiments. The dependence between the response and the alpha factor concentration, you can use one of these Hill equations with, of course, things with units, because the alpha factor is in nanomolar, it's not dimensionless like our IX. And in the case of frequency encoding, we sort of forced it to try to fit it in that way. But we found numbers that could reproduce more or less the observations of the experiments. And so this is the same I've just said. And for the model, what we have is everything is dimensionless. I mean, the time is not, but the stimulus strength is dimensionless. And so what we have is this. So we need to go from one to the other, from the dimensionless to the dimensionless stuff. And what we observe is that in the observations, in the experiments, H is equal to 1. And EC50 is between 3 and 5 nanomolar. And so we pick the curve with H equal 1, and we transform our dimensionless EC50 in a way that our 0.5, which is for this curve, could correspond to 3 or 5 nanomolar. That means making this transformation. And we did something similar for the case of frequency encoding. The case with asterisks, that's why we plotted them with asterisks, actually, is the case where the minimum time is similar to that of the experiments. And so and kappa is also similar. And so we only need to go from our dimensionless B to a dimensional B. And that's the transformation. And we put together the curves now in terms of alpha factor instead of the dimensionless stimulus strength, the two curves with asterisks of the previous slide. And what we see is that they cross over. So basically we think that maybe amplitude encoding starts to work at the beginning when the pheromone concentration is low. And then as the SHMU grows towards the region of larger pheromone concentration, the frequency encoding serves the purpose of allowing the cell to detect differences in concentration there in the region of larger concentration. So this is the end of the talk. So cells use different strategies to encode the environment. And sometimes the increasing concentrations result in pulsatile behavior of intermediaries of the signaling. In other times, it's not. It's more like a more one-to-one with some saturating thing in between. And so why? One possibility is this multiplexing. And here we try, well, this is part of a project in which we want to answer what's the advantages and differences or similarities between the two encodings. And here we computed the mutual information between the external stimulus and the messenger RNA accumulated. And what we observed was that the way in which they can discriminate stimulus strengths is qualitatively different between amplitude and frequency encoding, because amplitude encoding is like good to discriminate within a certain region, and frequency encoding can work equally well across a much larger range of stimulus strengths. And that derives from the scale invariant discriminating power that frequency encoding can endow the cells with. And so the apparently redundant combination of both mechanisms can serve the purpose of expanding the range over which the cell can react to the stimuli. And with this, I give you a message that I tend to give, and also make an announcement of a very interesting colloquium that will take place next Monday, which is the it's the World Quantum Day, and this is the International Year of Quantum, and this is something that we organized between IUPAP and UNESCO with Anne Lullier next Monday. So you have to register to be able to enter the Zoom. Hope that QR works, and muito obrigada.
Thank you very much. Time for more questions. We have one there.
Silvina, thank you so much. This is very exciting, and I think the first question, because I wrote down a bunch, but but I think the most interesting question I have is related to this trade-off between the amplitude encoding being able to distinguish very well this narrow range versus the frequency encoding being able to work across ranges. But as you said, the other processes in the cell ought to introduce some time scale constraint, and thinking that the cell needs to process information because it needs to adapt its behavior. It has to have the plastic behavior in response to these inputs. Do you think that there is a trade-off there? This trade-off is translated into a trade-off between able to identify something very well and respond to it quickly, versus being able to identify something more generally that you don't need to respond as quickly?
Yeah, well, it's an interesting thing that I hadn't thought about. But yeah, it may well be, in the sense that one is like needs some matching condition. But of course, we are thinking of only one gene or one transcription factor, so you can have many and maybe cover a region. But maybe it's not that efficient either. But so if you think of one thing, that would be, I think, an interesting, so that the amplitude is for fast, because frequency you need several pulses to be able to, so you need an observation time, which I didn't talk much about, but the observation time is important to count the pulses. And of course, the information increases as you collect more statistics, because you reduce the noise.
More questions? Nathan, I think.
So why can't you have both amplitude and frequency? Why can't the frequencies change? I mean, the different pulses
well, that's
have
something
different
that
amplitudes?
I don't know. It happened, of course. I mean, why the cells used both for the same stimulus? That's what you're asking?
No. I'm asking, so if once you have more than one pulse, you can change the amplitude on the different pulses, so it can do both at the same time.
Yes, we didn't drive that. The observations show, variations, because everything is very, variable. It's not so easy to reproduce things, because you need to collect a lot of statistics to draw conclusions in biology, because all cells are different, and so it's complicated. We didn't try to see that. There is, I think that also, I was thinking that maybe frequency encoding, which means you always go with the same sort of level of a transcription factor, can help finding the places where the transcription factor have to bind to. That's something that we haven't studied, but I think there is an advantage there because, I mean, if you go with, because transcription factors, they can bind to non-specific sites. And so if you send a bunch of them and many bind to non-specific sites, then some of them will find the right ones. So having a whole, range of, from small to large is not that good, I think. I mean, if you do everything to, because the pulsatile thing is not so simple. You need to go back to basal, and it's a lot of energy that you need to invest. Anyway.
More questions? If not, I can ask probably
Yes.
a long-term question. I mean, from what I understand, what you are doing is, let's say, build the small steps in order to understand, let's say, somehow the logic behind the cell and behind. When do you think, how many years it will be necessary to use this knowledge about the logic of the cell in practical studies, like in medicine or in biology? Or
Wow. I don't know about that. I mean, this is a very basic question we are asking. Why cells, I don't know, are using this strategy? But I really, when we started with this, I thought it was going to be that frequency was better because it was more resilient to noise. But then what type of encoding you put? And we put a stochastic encoding because there is something interesting about this stochastic thing, this exponential thing, which is that you have a continuous of frequencies, relatively large range of frequencies as a function of the stimulus. And while if you think of other types of transitions to a pulsatile behavior, which is, from the point of view of dynamical systems, is another type of bifurcation, which is goes to something that is with a very well-defined frequency, then it's like you didn't have the frequency, then you didn't have amplitude, and suddenly you start to have pulsatile with a fixed frequency. It's not that good to encode different stimulus. Here, the exponential is good to encode different stimulus strengths, but it goes at the expense that is stochastic. And if you wanted something non-stochastic, it would be less flexible for the. But the applications in medicine, I don't know. I really don't know.
Another simple question. Do you think, I mean, you showed this pattern for mutual information. Do you think other figure of merit goes in the same way, or is the only figures of merit that you can think, or?
Yeah, well, you can think, I mean, to detect gradients, you need other things also. I mean, usually detect differently in different regions of the cell and propagate the information. So there are other aspects to that. You can go into a more, this would be the simplest way, I would say, to approach the problem. But the detection of gradients, Herbie Levine, who is part of the physics of living system thing, he worked quite a bit on that. So how fast, I mean, how you propagate the signal, because you need to detect something in one region of the cell and something. Here we are thinking, well, whether it can discriminate or not, but there are other ways in which you can approach this, for sure.
Yeah, I was gonna ask about like the directionality of the signal as well. If these two different encodings could have some relationship on that, but I guess this it's a similar.
Yeah, well, yes, I guess so. Now we are going with a student, we are going to look at the experiments and also do a more mechanistic modeling of the tip moving of the, because the previous work that we did on that we didn't find much difference between amplitude and frequency, just different levels of sensitivity to the to parameters of the transcription. We started to think, well, if this, Alejandro said, when I see a cell like this, I know what's the pheromone concentration. And if I see like this, I know. And we were getting 1 bit, 2 bits, I don't know, mutual information. So we started to think of what the shape of the shape, something more related to space, which is not present here. And that's what we are entering now, trying to compute the mutual information between the shape and the pheromone concentration and do some modeling, which is a model that already exists. For the movement, there is like a patch here that sort of moves around and is more, it wanders less if it finds a large concentration of pheromone in a particular direction. So a more mechanistic kind of thing. That's something that we are doing now. And then, of course, I worked many years on, and I still do, on intracellular calcium signals. And there you also think not only of the frequency, but spatiotemporal behavior. And so that the information or the response that you generate does not just depend on the dynamics, but on the spatiotemporal dynamics. And for example, there are experiments in neurons where you do a protocol that induces a reinforcement of the synaptic strength. It's called LTP protocol, long-term potentiation, and then others that are long-term depression. And you can have different types of behavior depending on the spatiotemporal distribution of the intracellular calcium concentration. So what kind of response or end behavior you obtain not only depends on the dynamics of the signaling agents, but also of their space distribution. So there is more to it than time.
More questions? Yes,
So
apparently yes.
Yes, I have a last more question that I would be very interested in your opinion on that, which is this whole idea that allows us to bring physics and biology together, is this fundamental idea that evolution, well, ecological systems and biological systems must perform some tasks so well that they are driven to the limits of what is physically, mathematically, or chemically possible. In this case, you're studying the transmission of information. Do you think that in this particular example, in what types of information would be so important for the cell that the evolution would have driven it to the maximum of what is physically possible?
Well, you know, my biologist colleague, he's a little bit against that view. Well, because there is a lot of randomness in evolution. So I think he tends to think of the randomness more than, of course, then there is a selection afterwards, but it's by chance that you arrive at a particular solution. So maybe it could be very good compared to the previous attempt, but not the absolute optimal one. So he always is a little bit, he says, oh, no. He's against that, that view.
More questions? Okay, if not, we can thank our speaker again.
Okay, thank you.